Hi, everybody. My name is Utah Lee. I am the founder and CEO of Biosciences. I'm here to tell you about the human trophoblast stem cell. This is going to be a new starting stem cell platform that's available for anybody to use. So we are a pure stem cell platform company, Delaware C Corp, based in Philadelphia. We have full freedom to operate over these human trophoblast stem cells, so HTSCs. And my job is to get these cells to GMP, which I've done this month, and out-license it to all therapeutic companies to help them accelerate their therapies market. So as you guys all know, the biggest problem today with cell therapy is finding the right starting stem cell source. Ethical issues, inconsistent cell source. You have to choose between scalability and immunogenicity, and many times unclear FTO and CMC documentation issues. Well, we've solved all of these issues for everybody. Our cells have these characteristics. We can differentiate to all cells of 3 germ layers. They're ethically sourced, they're scalable, they're immune privileged, they're pathogen free, they have chromosomal stability to high population doublings, and we have a large IP portfolio to protect it. So here you'll see a continuum of fetal development. We get our cells, these HTSCs, from the embryonic stage, from ectopic pregnancies. These cells are mortal, and they're usually found between 4 to 8 weeks post-fertilization. On the left, you'll see a blastocyst, 5 days old. Our cells come from the trophectoderm lineage, so they're pre-placental. And what happens in an ectopic pregnancy is that the embryo is traveling down a fallopian tube and it gets stuck. And usually it'll stay there and keep growing until it bursts the tube. And if it bursts the tube, the mother may die of internal bleeding. It's actually the highest cause of death in first trimester pregnancies. And so what we've done is, when the mother feels a lot of pain, she'll go into the ER, gets scanned, the doctor will say, oops, you have a tubal ectopic pregnancy. They schedule a salpingectomy where they go in and ablate both sides of the tube, and they remove the mass and they discard it. And the reason they do that is because the embryo on the left side needs to implant into the endometrium by day 7 or day 8. But you don't discover the ectopics until 4 to 8 weeks after. So there are many weeks in between where that embryo has absolutely no connection with the mother, no nutrients going to the fetus, and it's understood by all OB/GYN that the fetus is non-viable. So what we do is we take the embryo and we scrape off the preplacental tissue, the chorionic villi, and we hand the mass back to the pathologist, we'll confirm it, and they still discard it. So we've effectively sourced discarded preplacental tissue pre-implantation. So these are probably the earliest naive cells that you can get without any ethical issues. We're doing 3 different programs. One is in Parkinson's disease. We're working with National Taiwan University. Our cells are very special in this way. We can produce neuroprogenitor cells in a 1-day protocol that is Th positive. And so we're putting them directly in a preclinical study with the National Taiwan University. These cells also, in a 1-day protocol, can produce pancreatic progenitor cells that is insulin producing. And some people ask, well, why is that, right? We don't know, but what we do know is theoretically, when the embryo is implanted with the mother's uterus, what happens when that mother takes a gigantic bottle of Coca-Cola? How does the embryo then deal with the hyper and hypoglycemia that results from it? Well, it's actually the placenta's role to kind of defend and protect the embryo from these effects, including pathogens. So our cells, very naturally, when you put a glucose response test to it, insulin will come out. So we're working with A-Star in Singapore to put together a program for type 1 diabetes, also preclinical. The NIH approached us last year to ask for the culture media because they wanted to characterize our extracellular vesicles for cell senescence. And you probably have heard these heterochronic parabiosis studies that was done at Stanford 20 years ago. What they did was they stitched two rats together, an old rat and a young rat, into one circulatory blood system, and it turns out that the young rat was getting older and the old rat was getting younger. So they did another experiment separately. They just had a plasma exchange, and it turns out that the same effect was taking place. So what does that really mean? That means that all of the factors that are kind of urging you to rejuvenate, become younger, sits in the proteins of the plasma. So the NIH approached us and asked us for all the culture media because we have the earliest cells that you can source to see its effect on cell senescence as well as reversing biological age. So we're working on that right now. Some of the first data will be published very soon and also released at a longevity conference in Dublin in June. So some of the characteristics, we're able to optimize our cells to grow out to 85 population doublings. And that's just using standard media, off-the-shelf media. So at 100 million cells per dose, that's 8 times 10 to the 17 doses. So we've effectively solved the scale problem. Our cells also naturally express HLA-G. So the embryo is a combination of father and the mother's genes. So how come it's able to implant itself into the mother's endometrium without her immune system going haywire and trying to reject it? Well, it turns out this HLA-G dampens down all of the mother's immune system and allows it to coexist with the mother. And so what we've done is 5 different animal studies, all with human cells in immune-competent rats, and all of the cells survive. We see homing and regeneration of defected organs, no teratoma or tumor formation, and we get behavioral results. And when we differentiate them to these neuroprogenitor and pancreatic progenitors, the HLA-G sticks. That means these cells are ideal for therapeutics. When we were doing the scale out to 85 population doubling, we did the last karyostat at 72 population doublings, very, very stable genome. And we believe that the reason why is because these cells have been literally incubated in the fallopian tube without any epigenetic effects from the microenvironment or going through the entire process of term, going through birth. So they're very naive and very early cells. We currently have 13 different GMP compliant lines sourced from Taiwan. And because we're in Taiwan, the cells, a third of the women that come in, third of the mother that come in have HBV, and almost all of them have CFV. And that's from testing the blood of the mother, right? So we took all the cells and we sent them to IDEX for the H-Impact-1 to test for 25 human pathogens, and they come back completely negative. So even though the mother has HBV and CMV, these cells are completely clean. So there's a defensive mechanism at the fetal-maternal interface that we don't fully currently understand. But that means our cells have a really, really clean starting source. Our cells are naturally adherent cells. So we did a small trial on microcarriers in suspension, and they grow really well. We also deployed a mRNA reprogramming method with FTO to drive them to an iPSC. And we took a standard kit, also retail, and drove to an NK cell and did a cytotoxicity study, and they kill like gangbusters. So we believe that it's because they are also very, very early naive cells, so they tend to be more active. We have developed a technology called GEMS, Gene Engineered Multi-Sites, where we literally have put parking spots in the genome. And when you use any gene editing tool, CRISPR, whatnot, you can put that plasmid right into that one spot and it will go nowhere else. We've effectively solved the random insertion off-targeting problem. So what we're currently working on with the former director of the NIH is a universal plug-and-play iPSC system. Right? And so the idea here is, imagine any immuno-oncology company that has a target. Now you can have that target put into the iPSCs, then differentiate them to all the various immune cells, NK, T cells, macrophages. Then you can set them against, in vitro, against the cancer target, And you'll be able to compare data apples to apples, right? Because the insertion only happens in one place. Whereas the current technology, there might be a lot of random insertions that you don't account for unless you sequence the whole genome. So this will be ready in about 6 months. We already have 50 patents granted and 47 in prosecution, all with Wilson Sonsini, 7 patent families. So the reason why we have so many patents and nobody has ever heard of us is because the lawyers advised us to keep building our portfolio and don't tell anybody that we have this until the market timing is right. And I believe that market timing is right now. Right. We have a wonderful team, management team, and really great scientific advisors. All of this data that you see here is done by a company in Seattle called Pluristics, with the exception of the animal studies, of course. So we have 2 platforms on offer, the human trophoblast stem cells. The RUO and GMP lines are available now. We also have an iPSC line that we created from the HTSCs. The RUO is available, and we intend to have the GMP done by the 4th quarter of this year. And all of the documentation is going to be done by Dark Horse Consulting. And so basically, what we have is a product market fit. We start with a platform with all the right cell characteristics. We de-risk the FTO issue with our patents and a CMC documentation issue with the help of Dark Horse. And we can provide scale. And because we are already at GMP, we can accelerate your product time to market and hopefully transform the current healthcare system and make cell and gene therapies really affordable to everybody. That's it. So if you have any interest in trying our new cell platform, please scan whatever platform you like or come talk to me after the presentation. And we'll send cells to you. Very good. Any questions? Okay. Oh, please. We haven't yet, no. Yeah, so we're working on type 1 diabetes, Parkinson's, and the cell senescence with NIH. So, if you want to work with T cells, whether from the HTSCs or iPSCs, you're welcome to it. The iPSC protocols and the HESC protocols do not work with the HTSCs. So it'll be kind of a black box to start with. But we already have HTSC direct differentiation to neuroprogenitors, pancreatic progenitors, hepatocyte progenitors, and we're working on kidney or renal cells now. Yes, please. Yes. They seem to stick when you do a direct differentiation to the various cell types, but if you turn them into iPSC, they do turn into canonical iPSCs, so they lose that. But we do detect the HLA-G intercellularly. Any more questions? Fantastic. Well, come talk to me afterwards. Thank you.